Welcome back to the Deep Dive, the place where we turn massive stacks of research into actionable knowledge.
Today, we are really handing you the blueprint. It's for one of the most complex, high-stakes journeys in modern science.
Talking about the transformation of a laboratory discovery into a functioning, market-ready health technology. This is about protecting a decade of your life's work.
That's really the core tension we're diving into today. We've been immersed in some fantastic source material, mainly excerpts from a researcher's guide to health tech patents. And the critical challenge, the one everyone talks about, is crossing the valley of death.
And that isn't just a catchy phrase.
Not at all.
Yeah.
It's not a metaphor, really. It's the treacherous chasm between a brilliant idea at the bench and navigating the incredibly complex, highly regulated landscape of federal law, the FDA, and intellectual property.
And our sources, they lay out a truly comprehensive strategy. We're going to cover everything.
We are.
This includes the innovation methodology itself, like how to actually invent effectively, all the way down to the granular details of federal oversight for both medical devices and for drugs.
And then there's the IP, the 5 pillars that determine if your work can even be patented in the first place.
And on top of that, the essential legal scaffolding that governs research funding, things like the Bayh-Dole Act and the American Invents Act.
So our mission today is really laser-focused.
Yeah.
We are trying to shift your perspective.
How so?
You can no longer approach this journey as sort of an open-ended scientific pursuit. It absolutely must become a highly disciplined, goal-oriented process of product development.
Okay, so it's a mindset shift.
Complete mindset shift. The singular focus has to be on creating something that maximizes both societal good and economic impact. When we're finished, you will possess a clear step-by-step blueprint for navigating that bench-to-bedside transition strategically.
Okay, let's unpack this, starting right at the genesis of innovation itself. You said we're moving past the traditional approach.
We are. The traditional and often, frankly, inefficient approach.
You mean the classic bench-to-bedside model?
Historically, yes. That discovery model started with a breakthrough, cool new molecule, fantastic algorithm, or some novel material discovered in the lab.
And then the researcher would spend years, maybe a whole career, just searching for a clinical problem that their discovery might solve.
Exactly. And while that has yielded some amazing breakthroughs, it's often a scattershot approach. It's a solution looking for a problem.
And contemporary innovation theory, which is exemplified by these highly successful models like the Biodesign programs at Stanford and the Texas Medical Center, TMC, it just fundamentally rejects that.
It completely flips the funnel. It becomes needs-driven. This approach asserts that a successful invention is not driven by the technology itself. It's driven by a meticulously characterized critical clinical need. If the need is robust, if it's real and well-defined, the resulting solution will inherently have relevance, market pull, and commercial viability.
So the process doesn't start with science. It starts with what? Observation.
It starts with pure observation. Let's talk about that crucial first step, clinical immersion.
Tell us about that.
Clinical immersion is an intensive period of pure ethnographic research. This isn't just about sitting in a library reading medical journals. You have multidisciplinary teams, and this is so key. You're mixing engineers, computer scientists, and researchers with the actual clinicians, and they spend literally hundreds of hours embedded within the healthcare system.
And I imagine they aren't just watching a surgeon operate. Where are they looking for the friction?
They are watching the entire workflow around the patient, from the initial diagnosis through the treatment phase into recovery, and critically, they are hunting for friction points in the administrative elements.
Like what?
Things like charting, data transfer, even billing. The objective is to identify all the pain points, the inefficiencies, the redundancies, and the outright failures in current medical practice.
So it's pure, focused observation, almost like being an anthropologist in a hospital.
It is. And the trainees in these programs, they were actively encouraged to collect hundreds of potential needs initially.
Hundreds.
Hundreds. And they have to withhold all judgment at this stage, which prevents them from prematurely filtering out what might seem like a small problem, but is actually a huge opportunity.
Can you give an example?
They might observe that a patient's feeding tube is really difficult to secure, or that the process of getting pathology results back takes way too long and delays treatment decisions, or that a specific surgical tool requires repeated, time-consuming sterilization between uses, which slows down the whole OR.
So why the mandate to gather hundreds? Why go for quantity over quality at the beginning?
Because the filtering process is where the true strategic value is unlocked, and you just need a huge sample size of problems to do that right. Once those observations are gathered, those hundreds of needs are rigorously filtered based on a whole matrix of criteria. You start with the technical, is it even solvable? But you quickly move to the market reality.
What are those key criteria?
The four crucial ones are the immediate measurable impact on patient outcomes, the size and accessibility of the affected patient population, the complexity of integrating a new solution into the existing clinical workflow,
That's a big one.
a huge one, and perhaps most strategically, the potential for significant cost savings within the greater healthcare system.
That last point feels absolutely critical. If the device saves lives but costs the hospital a fortune, it might never get adopted.
Exactly. But if it saves time and money, its adoption rate just skyrockets. You are prioritizing problems that the system wants solved, not just problems that are scientifically interesting. This filtering ensures the team is tackling a high-value target.
Which brings us to the pivotal deliverable of this entire stage, the concise, specific need statement. This is what anchors the whole invention process.
And this is where the intellectual discipline really comes in. The critical distinction, the thing you have to get right, is that a successful need statement must avoid embedding a specific solution.
Can't hint at the answer.
You can't. If you define the problem too narrowly, you severely limit the creative freedom you need for a true breakthrough in the next phase.
Give us that concrete example from the sources. I think that really helps lock in the understanding for the listener.
Okay, so a bad need statement, a solution-driven one, would be, we need a better surgical drill.
Right, that just limits you to making a faster drill or a sharper bit.
It's just incremental improvement. A robust biodesign needs statement, on the other hand, is defined as a way to prevent localized bone necrosis during high-speed orthopedic procedures in compromised elderly patients.
Wow, okay. That is a fundamental shift in perspective.
It is. That statement forces the team to rethink the fundamental mechanism of harm. It shifts the focus from optimizing existing hardware to asking these revolutionary questions. Is the issue heat? Is it rotational force? Is it the material composition?
Opens the door to solutions that aren't drills at all.
Exactly. It could lead to non-contact ultrasound or chemical softening agents or entirely new types of cutting instruments. You are solving the fundamental clinical challenge, not just iterating on an existing tool.
So once that need statement is bulletproof, defensible, and high value, we transition into the invent phase, solution mapping and iterative prototyping.
Right, so the team moves from that well-defined problem space to the open solution space.
Yep.
Brainstorming generates hundreds of potential solutions, but they are immediately subjected to this rigorous filtering in what the sources call a survival of the fittest mentality.
They're filtering against what?
Against technical feasibility, the existing IP landscape, and significantly preliminary regulatory risk.
So prototyping in health tech isn't just about building a cool model. It's inherently iterative, and it sounds like it's laser focused on risk mitigation.
Precisely. The early proof of concept models, they answer basic scientific questions. Does the underlying chemical reaction actually occur? Can the robotic arm achieve the necessary micromovement?
But in medical device development, unlike, say, general engineering, you're saying the regulatory layer has to be incorporated from the very earliest stage.
Yes, from day one, you have to be thinking about safety and materials.
So if your first alpha prototype is designed using a novel adhesive or some new polymer,
You must immediately ask, is it biocompatible? Will it leach toxic materials when it's implanted? If you don't incorporate those safety and biocompatibility requirements from the very first iteration, you just waste months, maybe years of development time and capital.
and the prototypes progress through different stages.
Rigorously, through alpha and beta stages, and each stage adds layers of complexity, incorporating functional requirements, ensuring ease of use by clinicians, that's human factors engineering, and confirming that it can be scaled for manufacturing. That regulatory mindset is just critical from the very beginning.
Okay, so once that robust concept or prototype is ready, the next gate you hit is the FDA. And that's where the path splits completely depending on whether you've invented a medical device or a pharmaceutical. Let's dive into section 2, the bifurcated path to market.
The distinction is absolutely fundamental because the evidence required for approval is so different. The FDA uses classification systems designed to match the level of regulatory oversight to the degree of risk posed to the patient.
And for medical devices, this is a 3-tiered system.
That's right. So let's start with the lowest risk tier. Class guide devices are low risk. They pose minimal potential harm. They only require what are called general controls.
What does that entail?
Things like proper labeling, adherence to the quality system regulation, and facility registration. The pathway is very straightforward, often just self-registration. We're talking about basic reusable surgical tools, tongue depressors, or oxygen masks.
Then we step up to class 2. This is the moderate risk category, and it sounds like this is where most novel devices actually land.
That's right. Class 2 devices require general controls plus what they call special controls, things like mandatory performance standards, specific labeling requirements, or maybe post-market surveillance. This is the sweet spot for many innovators, and they often use the 510K pre-market notification pathway.
Right, the 510K. And that relies entirely on a concept we mentioned earlier, substantial equivalence,
Substantial equivalence to a predicate device.
which is just a legally marketed device that's already out there.
Correct. It has to share the same intended use and similar technology. If you can prove your new infusion pump or your advanced glucose monitor is substantially equivalent in terms of safety and efficacy to one that's already been approved, the review is much more streamlined. It can save you years.
But I imagine this is a massive area for researcher pitfalls. I mean, how small of a change can push a device out of that 510K route and into something much more rigorous?
That's where the technical balance becomes so crucial. If you make a small change to the device material, but that change impacts the biocompatibility or the sterilization process, the FDA can say, nope, this is no longer substantially equivalent.
And what about the intended use?
That's even more critical. If you change the intended use, let's say you move a diagnostic tool from just monitoring to actual therapeutic intervention, that immediately nullifies the 5 and 10K path. It forces you onto a much costlier and time-consuming track, even if the underlying technology is very similar.
That rigorous track is class 3. These are the highest risk devices.
Correct. Class 3 devices are those that support or sustain human life, are of substantial importance in preventing health impairment, or pose a potentially unreasonable risk of illness or injury. They require the most stringent application, pre-market approval or PMA.
Describe the burden of a PMA. What does that actually look like?
Well, a PMA requires the applicant to provide what the FDA calls reasonable assurance of safety and effectiveness, and that has to be based on extensive clinical data.
So we're talking full-blown clinical trials.
We're talking multi-site, multi-year clinical trials involving large patient cohorts. The evidence must demonstrate a favorable balance of risks and benefits. Pacemakers, artificial joints, and certain types of complex implants all fall here. A researcher pursuing a Class 3 device, you have to understand you need to secure massive funding, and the timeline is typically double that of a 510K device.
And just to close out the device side, what if you have a moderate risk device, but it's completely novel? There's no predicate device anywhere.
That's the exact purpose of the de novo pathway. It allows the FDA to establish a new classification for a novel, moderate-risk device that falls outside the existing 3 classes. It gives you a regulatory path forward when substantial equivalence just can't be met, but the risk profile doesn't justify a full, expensive PMA.
Okay, let's shift gears entirely. The pharmaceutical path, which governs drugs and biologics, is less classification-based and more phase-driven, and it's notoriously long and rigid.
The journey starts with discovery, synthesis, and then intense preclinical research. This involves a ton of lab work and extensive animal testing to establish basic safety, metabolism, and toxicology profiles, all the crucial data you need before a drug can even touch a human subject.
That preclinical data is promising. The researcher files the gate-opening application, the investigational new drug, or IND, application.
The IND application is essentially a massive, comprehensive plan. It details the drug's composition, its manufacturing, and the initial proposed human trials. Once the IND is cleared by the FDA, then and only then can human trials begin, and they're structured into 3 very well-defined phases.
So tell us what Phase I is truly focused on.
Phase I is purely about safety and dosage tolerance. It involves a really small group, typically 20 to 80 healthy volunteers.
Healthy volunteers, not patients.
Usually, yes. Although sometimes patients with terminal diseases are included if the toxicity profile is expected to be severe. Researchers are trying to determine how the drug is absorbed, metabolized, and excreted, and establishing what's called the maximum tolerated dose, or MTD. Efficacy is secondary, or not even looked at here.
Okay, so if it clears that Phase I safety hurdle, you move into Phase II.
Phase 2 transitions to proof of concept and efficacy. Now, you are involving hundreds of patients who actually have the target condition. The objective is twofold. Continue monitoring for safety signals in the target population and gather preliminary data on whether the drug actually works.
And you're refining things here.
They're often refining dosage and administration schedules based on those early signals.
And then the definitive test, the one that consumes the most resources, is Phase III.
Phase 3 is just a massive undertaking. It involves thousands of patients, often split into randomized, double-blind, multi-site trials, comparing the drug against a placebo or a current standard of care.
And this is where you prove it works.
This is the definitive data set that has to statistically confirm the efficacy signals you saw in Phase 2, and you're meticulously tracking long-term adverse reactions. This is what proves the drug's therapeutic value and establishes its risk-benefit profile in the real world.
And success in phase 3 leads to the final regulatory submission. That's either a new drug application, an NDA for small molecule drugs,
A biologics license application, a BLA, for biological products like vaccines or antibodies. The NDA or BLA submission is a comprehensive dossier. It often comprises hundreds of thousands of pages.
wow,
It includes all the data from preclinical and clinical trials, plus rigorous details on the drug's chemistry, manufacturing, and controls, or CMC. The FDA review team spends months ensuring every single step, from raw material sourcing to final packaging, is compliant and safe. It is an incredibly resource-intensive road, and it's one that demands certainty about your intellectual property protection.
because no one is investing billions of dollars in a decades-long endeavor if that intellectual property isn't locked down. Which brings us right to Section 3, Intellectual Property Fundamentals.
A patent is the foundational asset. It really is. It is a legal right granted by the government, allowing the holder to exclude all others from making, using, selling, or importing the invention for a set period, typically 20 years from the priority filing date. Securing that right is just paramount to attracting capital.
And to be granted a patent in the U.S., you have to satisfy 5 statutory requirements under Title 35 of the U.S. Code. These are the 5 pillars every researcher absolutely must master.
Let's start with the first set, which is under Section 101, Statutory Subject Matter and Utility. Statutory subject matter just means your invention has to fall into one of 4 categories. A process, which is a series of steps, a machine, a concrete device, a manufacturer, which is a tangible article given a new form, or a composition of matter, like chemical combinations, even including GMOs.
But the courts have famously carved out judicial exceptions to this to prevent people from getting monopolies on the basic building blocks of science.
Precisely. You cannot patent abstract ideas, laws of nature, like gravity or E equals mc squared, or natural phenomena like a specific flower you just find in nature. And this is so crucial in life sciences, particularly in diagnostics and genetics.
So if your claim identifies a correlation between a naturally occurring biomarker and a disease, the USPTO, the Patent Office, applies the Alice-Mayo test.
Right. And let's spend a moment here, because this is where many modern life science patents fail. The Alice-Mayo test has 2 steps. Step 1. Does the claim relate to an unpatentable exception, like a law of nature? If yes, you move to step 2. Does the claim add significantly more to transform that exception into a patent-eligible invention?
So if I discover the law of gravity, I can't patent that. But if I invent a specific machine that uses gravity in a novel configuration to, power a device, I've transformed the concept into a specific application.
That's exactly the right analogy. The test is designed to prevent monopolization of the underlying scientific principle. Simply observing a correlation, saying patients with disease X have high levels of biomarker Y. That's not enough.
So what is enough?
You need an inventive concept that ties that observation to a concrete application, often involving a specific technological apparatus or a non-standard therapeutic intervention.
Coupled with that is the requirement for utility, which is also under Section 101.
The invention must be useful. But again, in health tech, useful is a very defined term. It must have a specific, substantial, and credible real-world utility.
And the key is that it cannot be merely a tool for further research.
Exactly. If your invention is only useful for discovering another invention, it generally lacks the required utility for a patent in the present day. You have to demonstrate it performs its intended purpose now.
Okay, moving on to the next 2 pillars. Novelty and non-obviousness sections 102 and 103, respectively.
Novelty is the easier concept. The invention must be new. It cannot have been identically disclosed in the prior art before your filing date.
And prior art is an expansive term.
Very expansive. It includes any publications, public uses, sales, or public knowledge available anywhere in the world.
So here's where it gets really interesting, I think. Even if an invention is completely new, it might still fail the patent test if it's not considered non-obvious under Section 103. And you said this is the highest hurdle for most innovators.
It really is the high bar, because it introduces this concept of the person having ordinary skill in the art, or posita.
This hypothetical person.
Hypothetical person. An invention is not patentable if the differences between it and the prior art would have been obvious to this posita. This person is assumed to have average skills and knowledge in that specific technical field. The Patent Office asks, would the posita have been motivated to combine existing elements to achieve this invention? And critically, would they have had a reasonable expectation of success?
It sounds incredibly subjective. How does a researcher prove that their invention, which often necessarily combines existing technologies, was in fact non-obvious?
They're relying heavily on what are called secondary considerations, or evidence of non-obviousness. The sources stress these factors can often swing a difficult case.
And what are those?
Include demonstrating unexpected results, that the invention performed better or differently than the prior art would have suggested it would. They include solving a long-felt but unsolved need, proving that others in the field had tried and failed to meet this challenge for years.
Or documenting the failure of others.
If you, the researcher, can point to historical attempts by well-funded competitors using different methods who failed to achieve the desired outcome, that provides powerful factual evidence that your solution was not just an obvious combination of known elements. These commercial and factual arguments are often more persuasive than the technical differences alone.
Finally, we reach the fifth pillar, which is essential for the actual drafting of the application: enablement and written description under Section 112.
This ensures the social contract of the patent system is met. You get a temporary monopoly, and in exchange, you give a full disclosure. The written description must be so clear and comprehensive that someone skilled in the art, the pasita, could reproduce and use the invention without undue experimentation.
So you have to provide the roadmap.
The full functional roadmap. Additionally, the inventor must disclose the best mode of practicing the invention known to them at the time of filing. This ensures that they don't hide the best way to make the product while patenting a less effective version.
Now, this entire complicated IP structure is being tested by the rapid integration of artificial intelligence into diagnostics and therapeutic guidance. How does the AI challenge shift the focus back to that abstract idea exclusion we talked about?
It's a huge, huge challenge. If your patent claim focuses solely on an AI model that calculates a risk score like predicting a patient's 5-year mortality risk, it will often be rejected as an unpatentable abstract idea or mental process.
Because you're just trying to patent math.
You're just trying to patent math. It's just computation.
So if the innovation is inherently in the algorithm, how do you successfully patent it?
The breakthrough must be anchored in a concrete, physical manifestation or a specific therapeutic action. The claim has to bridge the abstract AI output with a real-world change. For example, instead of patenting the algorithm that calculates glaucoma risk, the eligible claim would cover the steps of calculating that risk and then administering a specific non-standardized eye drop dosage based on that AI-calculated score.
So you're not protecting the mind of the machine, you're protecting the tangible system or the process that the machine controls and implements.
That's the strategic pivot. The claim becomes a system for treating X disease, comprising a processor that calculates Y variable and an actuator that administers Z dosage based on Y variable. The physical hardware and the resulting therapeutic change are what provide the patent eligibility.
That shift in focus leads us directly into the necessary strategies researchers have to employ to protect their work. Section 4. Strategic IP navigation. The move to the first inventor-to-file system under the America Invents Act, or AIA, made speed paramount.
Absolutely. Priority is no longer based on who thought of it first, but who filed the application first. And this immense pressure on timing is why the provisional patent application became the academic researcher's most critical strategic tool.
Describe the provisional application and its main benefit.
A provisional is basically an informal, low-cost 12-month placeholder. It lets you establish an early priority date without the immense rigor and expense of a full, non-provisional application. It doesn't require formal claims, oaths, or declarations, and you immediately get to use the commercially valuable patent pending designation.
And that 12-month window is a massive strategic advantage. It gives the researcher time to refine the technology, gather crucial efficacy data, and, most importantly, pitch the idea to potential investors or commercial partners.
It buys time and security. But there is a critical trap here, a huge one. The provisional automatically expires after 12 months, and it is never examined by the patent office.
Right.
A researcher might rush to file a provisional, believing it's just a rough draft. But to actually secure that crucial priority date, the written description in the provisional must be as complete and enabling as the final non-provisional application will be.
What happens if it's incomplete? What's the risk?
If the final non-provisional application contains details or embodiments, say a specific delivery mechanism or an optimized formulation that were not fully described in that initial provisional, the researcher loses priority for those crucial elements. They gain a false sense of security, potentially wasting a year, only to find that their effective priority date is the later non-provisional filing, opening them up to claims of prior art that came out in that intervening year.
That is a massive liability. Speaking of prior art, we absolutely need to address the researcher's existential conflict, the peril of public disclosure.
For academics, the pressure to publish or perish, conflicts directly and lethally with the requirement for patent novelty. Once you disclose your invention publicly, a poster, a talk, a paper, you have established prior art against yourself and that clock starts ticking.
Now, we do have the 1-year grace period in the US, which offers some safety net.
It does. U.S. inventors have a 1-year grace period after their own first public disclosure to file a U.S. patent application. But here is the critical fact that must be repeated over and over. Most foreign jurisdictions, including Europe, Japan, and China, follow a strict novelty rule.
Meaning, what does strict novelty mean in practice?
It means any public disclosure, even by one single day before filing, immediately and irrevocably forfeits your international patent rights in those territories. If your medical technology has global commercial potential, publishing before filing is just financial suicide.
Let's detail the specific disclosure thresholds because researchers often miss these nuances.
Okay, so a journal publication, print or online, instantly starts the 1-year U.S. clock and forfeits all your international rights. A conference presentation, either the oral delivery of the data or the distribution of a detailed abstract, has the exact same effect.
What about something like a thesis defense?
Critically, yes. Even a thesis defense is often considered a public enabling disclosure if it's open to the public or accessible through a university library or some online repository.
And perhaps the most dangerous pitfall in the modern age is the online preprint server.
Absolutely. Uploading a manuscript to a preprint server like RFEFT or BioRxF results in immediate public disclosure upon upload. It's an instant forfeiture of international rights. The standard operating procedure for every researcher must be: contact your technology licensing office, your TLO, before you even start preparing any public-facing material. TLOs can often file a same-day provisional application based on a draft manuscript or slides to secure that priority date worldwide.
So, what does this all mean in practice? The researcher's relationship with their institution is governed by this major piece of federal legislation, the Bayh-Dole Act of 1980.
The Bayh-Dole Act was the single most transformative piece of legislation for academic commercialization. Before 1980, the federal government retained ownership of any inventions resulting from federally funded research.
And that led to a huge disconnect.
A massive disconnect. Without the incentive of exclusive licensing and ownership, universities lacked the motive and industry lacked the mechanism to invest in commercializing those discoveries. Most of this promising federally funded tech just sat unused on a shelf.
Bayh-Dole changed the game by allowing universities and small businesses, the recipients of the federal funds, to retain ownership of the inventions and exclusively license them.
But this ownership comes with very strict government obligations. The researcher's first step is crucial. They must promptly disclose the invention to their university TLO. The university then reports that invention to the funding agency within 2 months via the federal iEdison system.
And the university has a 2-year window to elect title, which is basically deciding whether to assume ownership and pursue patent protection.
That's right. And if the university elects title, they are then mandated to file a patent application within 1 year and must demonstrate a good faith effort to commercialize the technology.
Researchers benefit directly too.
They do. The university must share a portion of the resulting licensing royalties with the inventor or inventors, which incentivizes disclosure. And on the policy side, any exclusive licenses granted by the university must generally require that the resulting product be manufactured substantially in the United States. This is a policy lever to ensure federal funding benefits domestic industry.
But the government doesn't completely surrender control. It retains some powerful residual rights.
That's right. The government keeps a non-exclusive, non-transferable, irrevocable, paid-up license for its own purposes. So they can use the technology internally, but they can't sell it commercially. The most important power, however, is the government's march-in rights.
March-in rights, that sounds like a powerful backstop.
It is. The government can effectively march in and require the university to grant additional licenses to other parties if the invention is not being made available to the public on reasonable terms, or if the technology is desperately needed to alleviate pressing health or safety needs.
Has that ever been used?
It's very rarely exercised, but the existence of march-in rights provides a critical policy assurance that federally funded essential technologies can't be withheld from the public arbitrarily.
Now let's circle back to the lab bench, because even with the AIA making the U.S. first inventor to file, meticulous lab documentation protocols are anything but obsolete.
Far from it. The lab notebook is still absolutely vital for several reasons. First, it's the primary source material for satisfying the detailed enablement and written description requirements of Section 112. Second, it is the only evidence a researcher has to prove derivation. That is, proving that a first filer actually stole or copied the invention from you, the true originator. The lab record is your legal defense.
So what are the protocols for creating legally defensible records? This goes way beyond just jotting down results.
Protocols are strict because they have to stand up in court. Entries must be made in permanent ink. in a bound notebook with numbered pages. Spiral bound or loose leaf formats are unacceptable because pages can be added or removed.
And entries have to be contemporaneous.
Right. Recorded as the data occurs. You cross out errors with a single line, never erasing or obscuring the original entry.
And the witnessing protocol is very specific. It's designed to ensure impartiality.
Exactly. Each page must be signed and dated by the researcher and by a witness. The witness has to be a third party who understands the technology conceptually, but is explicitly not a co-inventor or a co-worker. The witness isn't verifying the science, they are verifying the integrity of the record keeping and the fact that the documented invention existed on that specific date.
And you have to distinguish between the idea and the test.
Do. The record must clearly differentiate the date of conception, which is the definite idea, from the date of reduction to practice, which is the successful testing.
And for the increasing number of labs using electronic notebooks.
Electronic lab notebooks, or ELNs, must replicate that rigor digitally. They need secure timestamping, ironclad audit trails, and robust version control to prevent any unauthorized modification. A good ELN ensures that if you're challenged in litigation, you can prove exactly when an entry was made and that it hasn't been tampered with.
I've covered devices and general IP strategy. Now let's pivot entirely to pharmaceuticals and the competitive dynamics created by the Hatch-Waxman Act of 1984. This governs generic drug entry and patent protection. This is Section 5.
Hatch-Waxman is really the defining architecture for the modern pharmaceutical industry. It's a balance. It tries to balance the need to incentivize innovation by brand name companies with the public's need for affordable generics.
And the first component addresses the time lost to regulatory review. That's where patent term extensions, or PTEs, come in.
Right. The 20-year clock on a patent starts ticking when the application is filed, which is often years before clinical trials even begin. Hatch-Waxman allows a brand name sponsor to request a single PTE for the primary patent, the one covering the active ingredient.
And how is that extension calculated?
It's based on the length of the clinical trial and FDA review periods, up to a maximum of 5 years. It's meant to partially restore some of that lost monopoly time.
But the real strategic game begins with the mechanism for generic entry, the abbreviated new drug application, or ANDA.
The NDA is what allows generic companies to skip repeating the massive, costly clinical trials. They only need to prove their product is bioequivalent to the approved brand name drug. But the generic applicant has to legally address any existing patents listed for the brand drug by making one of 4 patent certifications.
And the certification that triggers the most strategic litigation is paragraph the 4th.
Absolutely. A paragraph the 4th certification is a high-stakes assertion. It's the generic applicant saying they believe the brand name patent is either invalid or will not be infringed by their generic product. It is a direct legal declaration of a challenge.
What happens when the brand company receives that notice of a paragraph 3 filing?
It almost invariably triggers a patent infringement lawsuit by the brand company. And here is the built-in strategic leverage that Hatch-Waxman provides. If the brand company files that suit within 45 days of receiving the paragraph 3 notice, the FDA is automatically prohibited from approving the generic drug for 30 months.
30 months. That's an enormous.
It's known as the 30-month stay, and it provides a critical period for the court to resolve the patent dispute. For the brand company, those 30 months are priceless. It allows them to maximize sales before competition hits the market.
But there's an incentive for the generic company to.
There is. The first generic company to file a paragraph 4th certification that successfully withstands the legal challenge is typically granted 180 days of exclusive generic marketing rights. It's a whole system built on strategic litigation and incentives.
This strategic leverage leads directly to the practice known as evergreening and the creation of these so-called patent thickets. This is where the tension between innovation and public access really heats up.
It is. While the primary patent covers the active chemical ingredient, companies frequently pursue secondary patents. These cover less fundamental aspects. Things like new formulations, say controlled release version, new dosages, specific methods of treatment, or even different crystalline forms of the active compound.
And these secondary patents are filed strategically, so they expire years after the primary patent would have run out.
The goal. So a generic company could be ready to launch once the primary ingredient patent expires, but they are still barred from market entry because their version of the drug would infringe on a secondary patent covering, say, the specific method of administering it.
So that is the patent thicket.
That is the impact of the patent thicket. And the sources cite some specific data here that is really quite striking. A staggering 91% of top-selling drugs maintain their market monopoly well past the expiration of their primary ingredient patent by relying on these thickets of secondary protections. Evergreening is a highly effective commercial strategy to extend exclusivity far beyond the initial 20 years.
That data point really crystallizes the complexity of the current system. Now, let's zoom out to Section 6, because all this complex strategic navigation, it doesn't happen in a vacuum. Successful technology translation thrives in these dense, specialized innovation hubs.
Ecosystem provides the crucial support structure. When you look at centers like the Houston Innovation Corridor and the Texas Medical Center, the TMC, you see environments that are explicitly engineered to shepherd high-risk health tech from concept all the way to commercialization.
And these hubs provide incredible structured innovation programs.
Yes. Beyond the TMC Biodesign program, which gives you that clinical immersion, they run specialized accelerators. The Accelerator for Health Tech supports digital health and medical device startups by providing critical access to the network of over 60 member institutions.
Which means immediate access to clinical testing sites, subject matter experts, and potential first customers.
Exactly. And for highly specialized fields, you see very targeted funding mechanisms, like the Accelerator for Cancer Therapeutics, which is CPIT funded and dedicated exclusively to translating highly specialized oncology discoveries into viable ventures.
Critically, these hubs offer technical infrastructure that academic labs rarely possess.
Like the Center for Device Innovation, the CDI at TMC. It provides access to industry-standard equipment, like ISO standard clean rooms, advanced CAD/CAM machinery, and specialized testing equipment for durability and biocompatibility.
And for a small team, that saves millions in capital expenditure and significantly accelerates the prototyping cycle.
It does. They don't have to build a commercial-grade lab just to refine a functional beta device.
And beyond the central hub, the institutional commercialization models themselves are adapting to be more efficient and industry-friendly. Let's look at some examples, starting with Baylor College of Medicine Ventures.
Right, so BCM moved to an internalized commercialization model, focusing resources inward to streamline the process. They reported a 42% increase in invention disclosures in the last fiscal year alone, which suggests that this dedicated internal support is significantly increasing researchers' willingness to engage with the TLO.
And a key feature of that BCM model that reduces risk is the Internal Target Validation Committee, or ITVC.
The ITVC is revolutionary for drug discovery. It functions essentially as an internal venture capital due diligence committee. It rigorously reviews academic drug targets using industry-level criteria, market size, competitive landscape, early toxicity data.
So they're triaging ideas early.
They're triaging scientific targets early, pulling resources away from purely scientifically interesting projects that are commercially doomed. And that dramatically improves the odds of successful therapeutic development downstream.
Meanwhile, the University of Houston Technology Bridge has optimized its IP process specifically to attract outside corporate sponsors with its preferred IP program, or P.
The PIPP removes the single greatest bottleneck in corporate academic partnerships, IP negotiation. It provides research sponsors with upfront certainty regarding licensing terms before the research even starts. By offering clear, preset licensing options, just reduces months, sometimes years, of legal back and forth.
Can you give us the specifics on those options?
Sure. For instance, their option B offers the sponsor an exclusive license combined with an upfront fee of 15% of the project cost, plus a modest 1% royalty on sales exceeding $20 million. Option C offers a non-exclusive royalty-free license for the company's internal use with a lower upfront fee. That level of transparency and clear framework drastically lowers the transaction cost for industry.
And finally, Rice University's Office of Technology Transfer, their OTT, emphasizes strategic planning right from the moment of disclosure.
OTT requires the immediate formation of detailed commercialization plans. Upon disclosure, they work with inventors to prepare high-quality marketing materials and, crucially, they mediate discussions with potential licensees to proactively avoid the conflicts of interest that often derail fledgling tech transfer efforts. It's a strategy-first, friction-reducing approach that respects both the academic and commercial imperatives.
This deep dive has really shown us that the transition from a brilliant scientist to a successful medical innovator is not accidental. It requires integrating scientific rigor with strategic foresight across IP management, regulatory affairs, and business development. It is a highly organized strategic campaign.
Indeed. The successful researcher of tomorrow has to think and act like a strategist, leveraging all these institutional resources like TLOs, the Edison system, and Biodesign programs, while maintaining a mastery of the legal scaffolding provided by the AIA, Bayh-Dole, and the complex mechanisms of Hatch-Waxman.
So let's reiterate the most surprising and critical nuggets we uncovered today for you, the listener. First, the high-stakes risk of public disclosure, particularly the immediate and irrevocable forfeiture of international patent rights if you publish that preprint or present that abstract one day before filing.
Second, the stringent yet absolutely crucial requirement of non-obviousness, that simply combining existing technologies isn't enough. You have to use evidence like unexpected results or the documented failure of others to meet the challenge to prove your invention's worth.
And finally, the strategic power of the 30-month generic drug stay triggered by a Paragraph 4 certification, a legal mechanism that provides brand companies with valuable monopoly time to maximize their returns before competition arrives.
And all of this brings us to our final provocative thought for you to mull over. We saw that specific data point that 91% of top-selling drugs maintain their monopoly well past the expiration of the primary ingredient patent. They do this because of evergreening via secondary patents on formulations, dosages, or methods of use.
The question we want to leave you with is this. Does the current regulatory and intellectual property framework, which was designed in large part 4 decades ago, still effectively balance the necessary incentive for pharmaceutical innovation against the public's need for timely, affordable access to essential medicines?
And think about this. If the bar for obtaining a secondary patent were subtly raised, perhaps through a stricter interpretation of utility or a higher burden of proof for non-obviousness as applied to these later patents, how might that shift the balance toward greater public access without destroying the financial engine that's required for developing the next truly breakthrough drug? That's the strategic puzzle facing both policymakers and innovators moving forward.